For over a century, biology has characterized oxytocin through a restrictive lens, focusing almost exclusively on reproductive physiology and acute social behavior. Recent integration of molecular neuroendocrinology and geroscience has provided a more complex view that moves beyond the simplistic cuddle hormone label. We define biological aging as a loss of physiological integrity, specifically driven by genomic instability and the failure of mitochondrial function. Within this framework, oxytocin operates as a central homeostatic pacemaker, maintaining cellular resilience across the mammalian lifespan. Scientists historically viewed the age-related decline of oxytocin as a benign biomarker of chronological time. Current scientific evidence now identifies this decline as a primary causative driver of systemic deterioration and multi-organ structural decay. This neuroendocrine collapse is driven by a transcriptionally repressive event at the level of the genome, rather than the irreversible death of hypothalamic neurons. Viewing oxytocin as an obligatory epigenetic geroprotector, a molecule required for genomic maintenance, provides a specific mechanism for reversing physiological frailty. The process begins with epigenetic drift, where physiological stress causes the oxytocinergic system to lose its youthful regulatory fidelity. This initiates a self-perpetuating cycle. First, methyl groups, 5-methylcytosine, physically silence the oxytocin gene promoter. This drop downregulates TET2, the primary enzyme for active DNA demethylation. Without TET2, the neuron plunges into an energetic deficit, downregulating mitochondrial complex 4. This forces the leakage of reactive oxygen species, increasing oxidative stress. Local stress is compounded by systemic inflammation. Elevated circulating markers like C-reactive protein and tumor necrosis factor alpha infiltrate the hypothalamus to reinforce the epigenetic silencing. These hypothalamic neurons remain physically present but exist in a functionally dormant state. This interconnected loop of methylation, bioenergetic failure, and inflammation represents the underlying engine of biological aging. Researchers have questioned whether this deeply entrenched epigenetic silencing is an irreversible terminal state. The Mejima study addressed this by testing a non-invasive intranasal delivery of exogenous oxytocin in aged murine models. The protocol involved a dosage of 10 micrograms administered daily for a brief 10-day duration. This chart tracks the systemic shift. Plasma oxytocin concentrations normalized to youthful levels, intersecting with a sharp drop in the inflammatory marker hsCRP. Rapid absorption of the peptide triggered a massive increase in neuronal activation within the paraventricular nucleus. Following the cessation of the 10-day treatment, plasma oxytocin levels remained elevated independently. The exogenous oxytocin reactivated the dormant hypothalamic neurons, allowing the brain to resume its own natural production of the neuropeptide. This intervention replaced the cycle of decay with a self-sustaining virtuous circle of endogenous molecular maintenance. The permanent genetic reset observed in the Mejima study is the result of a specific intracellular biochemical cascade. Oxytocin receptor signaling forces the TET2 enzyme to recruit a metabolic sensor known as O-glycanac-transferase, or OGT. OGT catalyzes the addition of a sugar molecule to histone H2B, a structural protein. This physically alters the nucleosome, forcing the tight chromatin coils to open. Once chromatin is accessible, OGT recruits the SET1/COMPAS complex. This complex applies the H3K4Me3 modification, a universal epigenetic marker for open transcription. Simultaneously, TET2 acts like a plow, oxidizing repressive 5-MC marks into 5-hydroxymethylcytosine and stripping the DNA of age-associated methylation. This coordinated action forces the cellular machinery back into a youthful and highly active transcriptional state. The TET2-OGT synergy functions as an epigenetic plow, clearing away the repressive modifications that characterize biological aging. The rescue of central oxytocin production exerts pleiotropic anti-aging effects, meaning it produces diverse, beneficial changes across multiple peripheral tissues. In musculoskeletal tissue, oxytocin targets muscle satellite cells via the MAPK-ERK pathway, driving the proliferation required to reverse sarcopenia, or age-related muscle loss. Within the skeleton, oxytocin acts on bone marrow mesenchymal stem cells, or BMSCs. It upregulates the protein RUNX2 to promote active bone formation. It simultaneously inhibits these stem cells from turning into fat and modulates the OPG-rank-L ratio to prevent the onset of osteoporosis. For cardiometabolic health, oxytocin upregulates PPAR-gamma to suppress macrophage inflammation, which significantly reduces systemic insulin resistance and the HOMA-IR index. The necessity of this system is demonstrated in genetic knockout models where mice lack the oxytocin receptor. These mice exhibit severe hyperphagic obesity, an uncontrollable drive to overeat, extreme metabolic derangement, and the premature graying of hair. Without continuous oxytocin receptor activation, the mammalian body remains vulnerable to rapid structural and metabolic collapse. Human longitudinal studies reveal a complex oxytocin trajectory that is deeply tied to evolutionary survivorship. Data from the Simane population of lowland Bolivia shows how oxytocin mediates evolutionary trade-offs in high-pathogen subsistence environments. Female oxytocin levels peak during primary reproductive years, correlating with the high metabolic demands of intensive childcare and breastfeeding. In males, oxytocin follows an inverse trajectory, remaining low in early adulthood, but peaking in old age to support somatic health maintenance. Older adults who maintain robust social networks demonstrate an increase in responsive oxytocin release compared to younger cohorts. High baseline oxytocin in the elderly correlates with preserved volumes of the hippocampus and amygdala, brain regions typically prone to age-related atrophy. Conversely, older adults who fail to maintain oxytocin tone show significantly higher systemic inflammation and elevated scores on the geriatric depression scale. Clinical translation of these molecular findings is already underway in human trials, such as NCT 020-69431. This trial investigates the administration of intranasal oxytocin to older adults to actively suppress inflammatory cytokines while preserving executive cognitive function. Engaging the TET2 epigenetic machinery provides a validated molecular mechanism to maintain biological resilience and extend the human health span.